In this video, we're really going to dig into the physiology of the nervous system, and this includes ions, channels, and diffusion. Some of this is going to be familiar because of the physiology that we went over in the skeletal muscle system. One thing that I would encourage you to do is I would encourage you to go back and look at your test 3 playlist and look at the generation of the resting potential and generation of an action potential videos. Okay, so the very first thing we're going to do is some definitions. And so voltage is the measure of potential energy that gets generated by separate charge. And this is measured in volts or millivolts. And the very first thing to say about this is basically that you've got one charge in one location and one charge in another location. And this is charge A in one location, charge B in a different location. And so your potential is going to be the difference between charge A and charge B. Now, the greater the difference between the points, the higher that the voltage is. So current is basically going to be the flow of electrical charge from one point to another. And current is something that can be used to do work because generally the flow of electrical charge involves the flow of some sort of particle. When you're talking about electricity in the wires in our home, you're talking about the movement of electrons. When you're talking about charge in the body, you're talking about the movement of ions instead. So this is something that can be used to do work, and the amount of charge that moves between, in this case, say charge A and charge B, is going to depend on two different factors. One is voltage and one is resistance. And so voltage, again, is the difference between those two charges. And the bigger the difference in between the charges, the higher that the voltage is. So now let's talk about resistance. And resistance is when something hinders the flow of this charge. So it's probably going to be a hindrance to the movement of these particles. And so this is, again, a substance through which the current has to pass. So an insulator is something that has high resistance, and you can see the red X that's right here between charge A and charge B. And so basically this is going to mean that this is going to cut down on the amount of current that's coming through. In case it can slow down, or in some cases completely stop the movement of charge between charge A and charge B, current between charge A and charge B. A conductor has got low resistance, and this means that the current is going to be able to easily flow from charge A to charge B. Sorry about that. I'm pretty bad at drawing arrows with a mouse. But anyway, a conductor is something that has low resistance and can therefore conduct a current pretty easily. Okay, so electrically excitable tissue means that the membranes of the tissue actually have current flow across them. And our two types of electrically excitable tissue are going to be nerve and muscle. So we've covered muscle in a previous test, and we're going to be looking at nerves. Now, there are two types of electrical signals. One is called action potentials, and action potentials can travel long and short distances. I don't want you to worry about this diagram too much. We're going to cover all of this kind of stuff in more detail. The second one is graded potentials, and these go for short distances. So one thing that I want to point out here is that you're usually going to find action potentials occurring along the axon of a neuron. You're going to find graded potentials happening in the cell body or in the dendrites here. So cell body and dendrites is where you're going to get graded potentials. So basically, good examples of graded potentials would be like right here. So there's a graded potential, and here's a graded potential, and here's another one. And what they can lead to, what graded potentials can lead to, are action potentials here and here. And we'll get into more of a discussion about that in this PowerPoint. So your transmission depends on what's your resting membrane potential. Is it at a normal resting potential? Is it above it or below it? But your resting membrane potential in any case is dependent on the presence of very specific ion channels that are located in the plasma membrane. And so as a reminder, ion channels are transmembrane proteins that allow the passage of ions. And these are basically a tunnel of ions through the membrane. And so basically ions that try to get through individual portions of the membrane here, they can't get through the membrane. The reason why is because they can't get through this hydrophobic portion in the middle of the membrane. So basically they're just turned away unless the membrane has a channel, and then the ion can flow through that either into the cell or they can flow to it outside the cell. So ions only travel through channels, and they do so down their electrochemical gradient. Now, I've got this in a couple of different colors here, and so the electrical portion right here, the electrochemical gradient, means that positively charged cations, and most of the ones that we're going to be talking about here in this PowerPoint are going to be positively charged cations. They move away from positive towards negative. So sodium and potassium are probably our two major players here. They're going to move because they're positively charged ions. They're going to move away from positive towards a negative charge. Okay, and this is what creates an electrical current that alters the membrane potential. But that's not the only factor that's involved. So there's also going to be a chemical gradient because there's going to be different concentrations on either side of the membrane. So they're going to move from an area of higher to lower concentration. Now I want to set up a scenario for you. Okay, so I'm going to have to apologize for my scrawling here, but let's say that this is a membrane. Okay, so that's a membrane, and let's say that you've got a very high concentration of potassium ions on the inside and a very small concentration on the outside here. So just a few ions on the outside. Now, in the meantime, let's put a whole big mess of sodium ions on the outside. Just bunches and bunches of sodium ions. So there's going to be a large positive charge out here compared to, since there are fewer positive ions, a lesser charge here. So let's just label these K plus here. And can't write at all with this thing. Na plus out here. Yeah, you ever sign your name on one of those like charge machines at a counter and you can hardly write at all? That's what this kind of feels like. So anyway, let's see what happens with the potassium. Now, if we were only paying attention to our concentration gradient, then potassium should flow from a higher to a lesser concentration. So you would see this electrical charge pushing the sodium out, right? So strong electrical charge pushing in that direction. But your electrical charge pushes back. And what this means is you only have a limited movement. You only have a limited movement of the potassium. So there's not going to be as much potassium moving out. Okay, and so sometimes the electrical and chemical gradients can work together, and sometimes they can oppose one another. Okay, let's talk about the different types of ion channels because they're usually closed unless something causes them to open. So the first type of channel we're going to talk about is leakage channels, and in leakage channels the gates are just going to randomly open and close, as you can see here. There's not going to be a particular stimulus that causes this. And as it turns out, there are more potassium channels than there are sodium channels, and because of this, the membrane is more permeable to potassium than to sodium. Remember that the membrane by itself without channels allows 0 ions to pass whatsoever. So the membrane can allow more potassium to pass than it does sodium, and this is what creates the resting membrane potential. Voltage-gated channels will open in response to a charge in the membrane potential. So let's say that something disrupts our membrane potential and moves the membrane potential to a certain point. At that point, when you have an electrical disruption, the voltage-gated channels are going to open. There's a different type, and these are usually involved in action potentials. Now, mechanically gated channels open or close in response to some sort of vibration. And so this can be sound waves or pressure or tissue stretching. So I want to make something clear to y'all. Voltage-gated channels will never respond to mechanical pressure. It's not what they're geared to do. Mechanically gated channels will never respond to voltage. It's not what they're set up to do. Okay, so a third type of channel is called a ligand-gated channel. And basically a ligand can be a neurotransmitter, it can be a hormone, it can be some sort of drug. But basically what happens here is that the ligands are shown here as blue triangles. So the ligand can bind to a plasma receptor, and then cause that channel to either open or to close. That's one thing that it can do. Another thing that it can do is that if it binds to this channel, then, or if it binds to the receptor, then the receptor can sometimes alter a protein that it's attached to, which might send a chemical message um, to an actual channel here that would then open and let potassium ions through. And I'm sorry, again, sorry for the scrawling, but that's the best I can do with this. So the ligand can bind to a receptor, and the receptor would cause, again, um, changes to an internal protein, which causes other changes down the line that allow, good Lord, it looks like a 2-year-old is writing this, doesn't it? I swear my arrows are worse than Donald Trump's signature. So it can allow another channel to open up and let sodium ions in. So again, the receptor doesn't have to be a channel protein. It can cause a cellular molecule here to open a channel protein. And examples of this are G protein coupled second messenger systems. Though you're not going to have to learn about those in this chapter. You'll learn more about those in A and P2 when you talk about the endocrine system. Okay, so this is just another diagram showing the ligand-gated channels opening when the neurotransmitter is bound to the receptor. So here you see this blank space right here. That's the space, that's the receptor, and you'll notice that it matches up to the shape of the neurotransmitter. So when the neurotransmitter happens, remember what happens when you bind or unbind something to a protein, it changes shape, and in this case the shape change results in the opening of the channel. Okay, this is a voltage gated channel, and right here I want you to notice on the left diagram you've got negative charges in here and positive charges out here, and on the other side here, look how the charge has changed. So you've had a disruption to the membrane voltage, and that disruption has caused the gates here and here to open here and here. Okay, so let's talk about how our resting membrane potential actually gets formed. So resting membrane potential is it's called the separation of positive and negative charges, but what it really comes down to is what I've got written in bold here. It's the difference in charge between exterior versus interior. So you can see here that the outside of the membrane is positive and the inside of the membrane here is at negative 70 millivolts. And so with neurons, your range can be negative 40 to negative 90 millivolts, but because by far the most typical value is negative 70 millivolts, then this is what I'm going to discuss. So with skeletal and cardiac muscle, the interior of the membrane is closer to negative 90 millivolts. Okay, so in resting membrane potential, you've got different ions that are involved in generating your resting membrane potential. So you've got different ions that are involved in setting up the difference between the outside and inside of the membrane. And so you've got some ions on the outside of the cell that are involved in this and some ions that are on the inside of the cell that are involved in this. And your main ions that are located on the outside are actually going to be sodium. Okay, so sodium is going to be on the outside. It is a positively charged ion. And chloride, which is also on the outside, but chloride is actually an anion, and so it's got a negative charge. Within the intracellular fluid, you've got potassium ions, and these have a positive charge, and you've got organic phosphate and proteins, and most of these have got a positive charge as well. Okay, but the next thing that I'm going to tell you is that your major players here are sodium and potassium. So everything else has got such a small role that we generally don't consider those very much. So we're mostly going to consider sodium and potassium when we talk about any of these potentials. So the way that this was discovered, the difference between the inside and the outside, was that some really good work was done by physiologists in which they were able to place a microelectrode here on the outside of the cell and place another microelectrode here on the inside of an axon. And so your voltmeter went to 70 millivolts. And so that meant that the charge on the inside was 70 millivolts less than the charge on the outside. So I want you to remember that whenever you're talking about potential, you are always talking about the inside of the cell. when you talk about the membrane potential being negative 70, it's always negative 70 on the inside of the cell as compared to the outside. And so if you see a rise and a fall, it means that something is happening on the interior of the membrane to cause this rise and fall of the membrane potential. Okay, so your main ions that are involved are, as I said before, potassium and sodium. And so your membrane is 50 to 100 times more permeable to potassium than it is to sodium, and it means that more potassium will leave the cell compared to the inward movement of sodium. And so this means that what I should really tell you is that when you're talking about cell membrane, I'm going to draw it right up here in this upper corner here. So that is supposed to be a membrane. There is a lot more potassium on the inside. I really wish I could do this more neatly than there is on the outside. So way more potassium on the inside than there is on the outside. That's supposed to be a very small K. And then on the outside, there is way more sodium on the outside than there is on the inside. So there are some big time concentration gradients to begin with on the outside versus the inside. Now if you've got channels open for potassium, that means that it's going to tend to flow outwards, okay? And if there are channels open for sodium, it means that there's going to be the flow of, excuse me, got the wrong color there. There's going to be a flow of potassium inwards, okay? So sodium flows in, potassium flows out. Okay, now overall, there are more sodium ions overall than there are potassium ions. And so with fewer ions inside the cell, even though sodium and potassium are both positively charged, as you can see from the pictures here, from here and here. So look, sodium is positively charged, potassium is positively charged. Even though both of them have positive charges, there's more ions on the inside, on the outside, than there are. I'm sorry, the charge inside the cell is negative because there's fewer ions inside the cell than there are outside. And so here's an analogy that I can give you, and I think I gave this to you with the skeletal muscle physiology. So if I have $30 and you have $100, we've both got positive money, okay? I've got plus 30, you've got plus 100. But compared to you, I have negative $70. So even though there's positive potassium on the inside and positive sodium on the outside, compared to the outside, the inside has negative 70. Okay, so you maintain your membrane charge in two different fashions. One is diffusion of sodium and potassium across the membrane, and this is dependent on both the concentration on the outside and the inside and what's happening with the charge on the outside and the inside, and the sodium-potassium ATPase pump. And so we'll get into this in a little bit more detail. Okay, so the first thing to say is that the permeability of sodium and potassium across the membrane are different. But there are some things that I want to do here, okay? If a cell only has potassium channels, and we put in our channels here, and we put in our ions, there's going to be some migration of these potassium ions from the inside of the cell to the outside. So as the positive ions leak out, then this voltage on the inside that is negative will tend to try to keep those ions on the inside. So I want you to recall that compared to the outside, this interior has got a negative charge, and that's supposed to be a minus mark. So a negative charge right here inside as compared to the outside. I sure wish I could type with this. So this kind of pulls the positively charged potassium ions back in, and what I want you to notice is that the flow gets slower as it goes on, because eventually there's going to be so much more of a positive charge on the outside that it will not allow any more of these positively charged ions to exit. And so in this case, now I want to make it clear there's still way more potassium on the inside than there is on the outside. I just couldn't draw all that in. Even though some of the potassium has exited, even though if it was just up to concentration gradients, more would exit. It can't, because there's now there's such a strong positive charge on the outside that that is prohibiting these positively charged ions to actually leave. Okay, so the next thing that let's do is um, let's add in some positively charged sodium ions, and so way more on the outside than there are on the inside. When you open up these channels, then a few of them will move from the outside to the inside. Okay, so what that's going to do is that's going to actually drive our our membrane potential back towards a more negative direction. And so what happens is the balance between all of these things is going to balance out at negative 70 millivolts. Okay, now how do you keep this stable? That's done by the sodium potassium ATPase pump. Now what this is going to do, so here's a picture of the pump, is it's going to actively transport 3 sodium ions out per turn and 2 potassium ions in per turn. So basically what it's doing is it's kind of opposing the actions a little bit of these leak channels, and it's helping to stabilize this resting membrane potential at negative 70. So again, this is all about stabilizing our resting membrane potential at negative 70. Okay, now if you have a disruption to that negative 70 value, then you can get an action potential started. And so an action potential involves a completely different set of channels in the plasma membrane. So let's start out by saying this. There are leak channels everywhere on the entire neuron, in the axon, in the cell body, in the dendrites, everywhere. Voltage-gated channels are only located at the axon and the nerve terminals. And what that means is that only the axon can carry an action potential. So you can only have an action potential located in the areas where you have voltage-gated sodium and potassium channels. Now, if your resting potential of the membrane gets disrupted, that disruption in charge will allow first the sodium channels to open, then the potassium channels to open. Now, these action potentials are an all or none phenomenon. So the threshold happens when your membrane potential hits negative 55. Negative 55 millivolts is your threshold. If you're at negative 56, nothing happens. When you hit negative 55, the whole thing happens. The action potential, the whole event of the action potential happens. So it's like a light switch. It's all the way on or it's all the way off, and there's no in between. Now the action potential will always be the same size or amplitude, and that makes a little bit more sense when you look at a graph of it. Okay, so here's a graph that shows this, and what you can see here is on the y-axis you've got a charge here of negative 70 millivolts. Okay, that's your normal charge. Now this right here, the dotted line, is the threshold at negative 55. If you have a disruption that pushes that charge up to negative 55, then you're going to see an action potential start. You'll get these phases called depolarization, repolarization, and hyperpolarization occurring. We'll go into more detail about that in just a little bit. And then from hyperpolarization, you see some things happening that return us to threshold. And again, I'll explain what those details on later slides. Okay, so let's talk about the depolarizing phase first. Now, the first thing to tell you actually is that, let me remind you of something you may have experienced in your past, in your distant past. So it certainly is a distant past for me. So let's say this is a bar magnet. Pretend that's a rectangle, and you've got positive and negative poles on that magnet. That magnet is said to be polarized. Now, if you consider a cell membrane, you've got a positive charge on the outside and you have a negative charge on the inside. And so the membrane is said to be depolarized. And so what happens in depolarization is that the resting potential moves away from negative 55 up towards 0, and in fact moves past 0 all the way up to positive 30. Okay, and what has happened is you've actually gotten rid of your poles, and so that's said to be depolarization. In repolarization, you actually have the resting membrane potential restored. And so the resting potential um, moves from here back down to -70. And it's during this portion of the action potential that you have repolarization. Okay? Now, this action potential is caused by changes in the permeability of the plasma membrane. So I want you to see what's happening here. Depolarization here is caused by changes in sodium permeability. How do you change the permeability of sodium, or the permeability of the membrane to sodium? Well, the only way that you can do that is to open up sodium channels, right? Remember that permeability is a measure of how many things can pass through a membrane. So the only way to increase sodium permeability is to increase your sodium channels. And the reason why you have this quick increase in sodium permeability is because this is when your voltage-gated sodium channels open. So depolarization happens when your sodium channels open. If you'll look at if you look at repolarization, during repolarization you have increased potassium permeability, and the same sort of principle applies here. You open up more channels to potassium, then more potassium can actually flow out of the cell. And so that's why repolarization, where your charge drops, occurs when all these positive potassiums leave the cell. So I want you to notice something else here about the potassium permeability. It has a sharp tickward up, and it has a sharp tick down. And this sharp tick down happens when the sodium channels get inactivated. So what's happening here? Sodium voltage-gated channels open here, your charge goes way up, then right here they inactivate, and then your charge goes back down because close to this same time, your potassium channels are opening. Okay? So when sodium moves into the cell, you get more positive from positive ions moving into the cell. When potassium leaves the cell, then you have positive charges leaving the cell, so the enzyme becomes more negative. Now your fourth stage here is called hyperpolarization. You've had so much potassium enter the cell that not only has it dropped to your original -70 value, but it's actually dropped beneath it. And so now the cell isn't just polarized, it's hyperpolarized. Okay, so again, depolarization, voltage-gated channels open, and your plasma membrane goes from -55 up to +30. So let's see some things here. With this voltage-gated sodium channel, you've got two gates. So it's a little bit more complex than other channels. You have an inactivation gate here, and you have an inactivation gate here. And this is what the sodium channel looks like at rest during resting potential. So let's take a look at, again, at this channel at rest. So at the resting membrane potential, the inactivation gate, this is the activation gate, it's closed, and the inactivation gate here is open. Now when you go to threshold, when you hit threshold, then one thing happens. Take a close look at the activation gate, that's the gate in green, and watch what happens. It opens up. Now at this point, when you hit -55 at threshold, these sodium channels open up, and now sodium is able to come through, and it comes through until you hit +30. Now when you hit +30, you have another shape change with this protein, and what happens, if you'll watch the inactivation gate at the bottom, it closes up. Now this gets called the inactivation gate because when this gate carries out its function, it inactivates the sodium channel. So as long as your inactivation gate stays closed, there is no passage of sodium at all through these channels. Now sometimes late during repolarization, the inactivation gate will open back up, but the main time period during which the inactivation gate reopens is during hyperpolarization. And during hyperpolarization, the activation gate closes, so you'll notice that that green gate now is now closed. The inactivation gate is now open, and if you think this looks just like resting potential, well, you're exactly right. This is a reset to the resting potential state of the sodium channel so that you can have a second action potential. Okay, now voltage-gated potassium channels, they also open at threshold, but they open way more slowly. So they don't come all the way open until you hit positive 30. So at positive 30, you see, now if you look at the sodium channel, you'll notice it's only got one gate. And at positive 30, that gate opens, potassium rushes out, and it's at this point that you see the beginning of repolarization. And so with repolarization, you move from plus 30 back down to negative 70, and these voltage-gated potassium channels will still remain open. Now they're going to close during hyperpolarization so that you get back to your original state. So unlike sodium channels, which can be open or closed or inactivated, voltage-gated potassium channels are either closed or open. Okay, and again, you can see this is what it looks like at hyperpolarization. You return basically to the resting state. Okay, so during the hyperpolarizing phase, most of the potassium channels are still open. They do close during this time, but they close more slowly than the sodium channels do. So your plasma membrane charge can have a charge of up to negative 90 millivolts, but at that point, those leak channels start kicking back in and working, and so does the sodium-potassium pump, and it'll return the membrane potential to negative 70 millivolts very quickly. Okay, now a refractory period is a time during which an action potential cannot start in a cell. Okay, so the cell can't start a second action potential in the middle of the first one. So there's something that's called an absolute refractory period, and this is when a cell cannot generate an action potential no matter how strong the stimulus is. And this occurs during the time when the sodium inactivation gate is closing up the channels. Okay? Now, a relative refractory period means that you can generate a second action potential, but only if you've got a larger than normal stimulus. So this occurs while the voltage-gated potassium channels are still open and the sodium inactivation gates have returned to their resting state. So, okay, so let's take a look at what happens during the action potential. Let's take a look at the absolute and relative refractory periods. So again, the absolute refractory period is when you cannot start another action potential, and the relative refractory period is when you can start an action potential, but it takes a stronger stimulus. So let's go all the way through the action potential first, and then we'll discuss it. So right here is resting membrane potential. Now, the voltage-gated channels are in the resting state. Take a close look at that. Resting state means the activation gates are closed, the inactivation gates are open, resting state. Now, you can open up the channel from the resting state. Okay, so what do you do to open this channel? You have some sort of stimulus happen. So as long as the stimulus has not reached threshold, then the sodium channels, again in the red, stay at their resting state. The activation gates stay closed, the inactivation gates are still open. At this point, also the potassium channels are still closed. So let's go into depolarization. So here's our depolarizing phase. Now look at what's happened to the sodium channel on the left. The sodium channel has now opened. It has its activation gates open. It has its inactivation gates open. It's all the way open. Sodium can pass through. Now this begins our absolute refractory period. You can't, basically the beginning of an action potential, it starts when you open up the channels. You can't open the channels if the channels are already open. I hope that makes sense. It's like you can't open a door if it's already open. So this is an absolute refractory period. As this portion of the action potential is going on, you can't start another one. So here's our current state of events. Between -55 and +30, sodium is all the way open, potassium is still closed. And let's go into part of the repolarizing phase. So during this part of the repolarizing phase, look at what's happened to our sodium channels. The sodium channels are inactivated, meaning that the inactivation gate has closed. So as long as the sodium channels are all inactivated, you cannot start another action potential. You can't start an action potential directly from this arrangement of the sodium channel. Also note that the potassium channels are open. So this is the second portion of our absolute refractory period. You cannot start a second action potential so long as these sodium channels remain in activation. So the next thing I want to do is ask you a question. So eventually these sodium channels are going to return to their resting state where that inactivation gate opens and the activation gate closes. Do you think they're all going to do that at exactly the same time? The answer is no. They're going to open at different times. So they're not going to open in exact unison. At some point you're going to have like 5% closed, 95% inactivated, 10% closed, 90% inactivated. So if you're 10% closed and 90% inactivated, it means that 10% of the channels are available to start a second action potential. And so the more of the sodium channels that return to the resting or closed state, the more that return to the resting state, the weaker the stimulus it takes to open them all back up. But that's still going to be stronger than if all of them are in the resting state. So let's say at a certain point in this repolarization, you have 50% resting state, 50% inactivated. Hopefully it makes sense that at that point it will take a stronger than normal stimulus to start a second action potential. But the point here is we are now at this point in the relative refractory period where some of the sodium channels are closed, some are inactivated, and so you can start a second action potential. It just takes a stronger signal. Okay, so also notice that the potassium channels close during this time. They do start to close during this time, but they close more slowly than the sodium channels return to their resting state. Now the final stage here is when everything is completely reset. So during that sort of lighter tan color and during the green color, this is your absolute refractory period. You cannot under any circumstances start a second action potential. During the darker yellow portion, this is a relative refractory period where you can start a second action potential, but you have to have a stronger stimulus. Now, if you're all the way over at the left, only a few sodium channels have returned to resting state. It's going to take an extremely strong stimulus to get a second action potential started. Let's started like right here. So maybe right here only 5% of the sodium channels are at resting. Maybe right here 50% of your sodium channels are at resting. And maybe right here 99% of your sodium channels are at resting. So it's going to take a really, really strong stimulus to open your channels here, or to start a second action potential here. Stronger than your normal stimulus, but still weaker than the left-hand stimulus to start here. And here, a stimulus here would only have to be slightly stronger than under resting circumstances, because here 95% have actually returned to that resting state. Okay, so the further that you go along in the relative refractory period, the easier it becomes to start a second action potential until you hit this phase, resting potential phase again, at which point a normal stimulus can start it. So here's our summary. Okay, so again, the sort of peach colored and the darker amber colored periods are the periods of your refractory period during green and peach, absolutely no possibility of a second action potential. During the amber portion, as you move to the right there, then it becomes easier and easier and easier to start a second action potential until when you hit the purple again, you can start a second action potential with a normal stimulus. Okay, so again, this is just a second depiction of that, and you're going to have an absolute refractory period in the green portion and a relative refractory period in the darker blue portion.